system, we have to have a method. And that method is protein docking. Now, this particular docking method is nothing but it is a bioinformatics or computational tool. This tool is used to identify the possible binding regions and the possible binding conformations between two proteins or a protein and its ligand. There are a multiple number of wet lab techniques to identify the protein structures. Can I have the next slide, please? Next, yes. See, this is in a nutshell how a docking is performed. This is the receptor and this is the ligand. R stands for the receptor. Can you press another enter? No, just the back one, Yeah. The R, the receptor, and L is the ligand. Receptor is basically the protein that we are interested. And ligand that I mentioned earlier, that it can be anything. Ligand fits into the cavity of the protein. Here, this is in a nutshell how a protein docks to its ligand. The next slide, please. The importance. Now, why should we perform docking studies? The docking studies are performed in order to generate the protein complex structures. Now, what is the importance of a protein complex structure? As I told you earlier, that protein complexes play vital roles in most of the biological processes. That is why we need to identify their possible binding interactions. Press and enter, please. Yes. We can understand also how a protein network operates. What is meant by a protein network? A protein network means it is a multiple number of proteins, a combination of proteins. That means, say, in a particular disease, say a cancer. Cancer means in this, you all know the basic mechanism of cancer. Cancer is the unrestricted growth of a particular tumor. Now, the unrestricted growth of the tumor is mediated by interactions between several proteins. So in order to know how one protein interacts with the other and how that particular interaction leads to the overproduction of certain other substances, which leads to the amplification of the signal to proliferate a particular tumor tissue, that is called the networking. So this networking can also be studied with the help of docking. Press and enter another. Now, this is another very vital thing that that I was talking about a few minutes back, that thermodynamics and molecular recognition. As I told you earlier, that thermodynamics plays very, very vital roles in our biological systems. So from the docking studies, we could identify how that particular protein complex, how much it is stable. And a stable protein complex would signify that that protein complex has certain biochemical actions, functions. So that thermodynamics aspect of a protein complex formation is also very important, and that can be studied with the help of docking studies. The next slide, please. Yes, this I will talk about later. The next slide, please. Yes, the thing that I was talking a couple of minutes back is the various types of tools. Here, I would like to mention wet lab, because in some cases, it is expressed as experimental tools. I have strong objection to that, because bioinformatics is also an experimental biology. So We cannot say it is not an experiment. It is a computational experiment, and the other one is the wet lab based experiments. So there are wet lab based techniques or wet lab based tools to identify the protein complexes. What are the wet lab based techniques? These are the X-ray crystallography and NMR. These are the most important ones, very vital ones. And we get lots and lots of information from this. Then there is the East-2 hybrid system and binding affinity calculations. But the problem with these wet lab techniques are that there are lots and lots of problems. But what I should mention here is that these wet lab based techniques, they give you the real life picture of protein complexes. Definitely, there is no denying in it. But all these wet lab based techniques, they are very, very expensive. And they are they are dependent on lots of factors. The manpower, it is labor intensive as well, and there are lots and lots of failures. So, and as you all know that these days, most of the labs are closed. So how will you perform the wet lab based techniques? So the only alternative is the bioinformatics. Only alternative is bioinformatics. Now, the thing is that that means how accurate the bioinformatics docking procedures are. They are fairly accurate. I'm not saying they are 100% accurate. It will be a misconception. I would lie if I say it is 100% accurate. It is not. It is impossible to have 100% accuracy, but they are accurate enough to mimic a cellular system. At least from these docking studies, you can have some amount of idea about the possible binding interactions. Okay, the next slide, please. Yes, this concept, I would like all of you to take good notes on this, the thermodynamics of binding. So, what is important here is that the most important aspect is the delta G. Delta G means it is the binding free energy. Now, what is meant by binding free energy? We all know what a free energy is. Free energy means it is the amount of energy that is being released or used up to perform any work. Now, for any spontaneous process, delta G has to be negative. And the value of delta G can be calculated in this way, where RL stands for concentration of receptor ligand complex, and R and L determine the concentrations of the receptor and the ligands respectively. But the most important one, the delta G should be negative. Whatever the case may be, the delta G should be negative. Can I have the next one, please? Yes. Press enters. Another. Yes. Now, the types of docking. There are two modes of docking. One is called the bound docking, and the other one is called the unbound docking. Now, what is bound docking and what is unbound docking? In case of bound docking, sometimes it is possible that we know the complex. We have the structure of the complex. From there, what we can do is we can separate the two components, the receptor and the ligand. And we perform certain mutations in the receptor, or certain mutations in the ligand, or certain mutations in both. And then we let them bind together by the docking procedure. This is done in order to identify that how these particular substances, that means the protein and its ligand, how they behave when the amino acids are mutated, whether they are behaving in the same way or not. And this would also give us an idea about which amino acids would be functional and which amino acids are non-functional. See, say in wild type complex, say amino acid X interacts with amino acid Y of the ligand. Now, if we mutate the amino acid X and try to identify how it interacts with amino acid Y of the ligand, and if we find that after mutation, no interaction occurs, in that case, we could say that, okay, amino acid X is very much important. This is called the bound docking, a very important aspect. And the unbound docking, we can here we know the structures of the complexes in case of bound docking. But in case of unbound docking, what we know is that we know the structure of the individual components, but we do not know the structure of the whole complex. In that case, what we can do is we can let them get docked together. And whenever they get docked together, we can identify the structure. Now, in all the types of docking procedures, we get numerous structures, say at least 10 different complex structures or 10 different conformations. So which one to choose? Because we don't know how the individual components interact. Here comes the energy minimization. I think I discussed about energy minimization in my previous lecture. And energy minimization is basically the process to identify the lowest energy conformation of any system. So among the 10 different conformations, we use the energy minimization technique. We run the energy minimization techniques on all these 10 different conformations of the receptor ligand complexes and identify which one has the lowest energy conformation. From that, we could predict, okay, this one, say we have A, B, C, D, E, F, G, H, all these conformations. And after energy minimization, we find that, say, G has got the minimum energy. So we can consider that among all these conformations and between A, B, C, D, E, F, G, G has got the minimum energy and G is the most probable conformation of the docked complex. Here, this is a guess because we do not have any idea about the structure of the docked complex. In that case, this is a good guess. In most of the cases, I would say in 90% of the cases, we could predict the biological processes using this method, this unbound docking method. The next slide, please. Next slide, please. Okay. Press and enter. Yes. Now, a little bit of algorithm that we must be familiar with, I should say. In this case, what we like to have is a binding site information. A binding site information means this is the information we get to know from literature that, okay, we know these portions of the receptor and these portions of the ligand are known to be interacting with each other. Now, what we can do? We can use this particular interaction while docking the two substances. This particular information, using this particular information, we can generate as many as 100 or 1,000 or as many as a number of different conformations. And then the procedure is the same. We perform energy minimization. And from the energy minimization, we could predict the conformation of our interest. The next slide, please. This is the post-processing I mentioned earlier. So the next one. Now the energetics. Here, the energy minimization. The energy minimization, I told you, it is the process to get the minimum energy conformation of any particular substance. Now, in order to perform the energy minimization, what we need to know is that, excuse me, we need to know what type of binding interactions or what type of delta G values we need to consider. The overall delta G is the consists of the van der Waals interaction, the desolvation, the electrostatic interaction. And another one I will explain. I'll explain each of the terms separately. Now, let us first consider what is van der Waals interaction. Van der Waals interaction means this is the interaction which is operative between the non-covalent portions of the receptor and the ligand. We all know that in most of the biological molecules, there are non-covalent portions, and that means the hydrophobic portions. Those hydrophobic portions, they interact using van der Waals forces. Then desolvation. Desolvation means how much energy is released when you try to dissolve them in a particular solvent. Electrostatic. This is another very vital phenomenon because in most of the proteins, we know you have the CO and NH groups. And in case of CO, the oxygen is negatively charged. In NH group, nitrogen is positively charged. So there is an interaction between the negatively charged oxygen and the positively charged nitrogen. That is called the electrostatic interaction. And the last one is whenever we are generating the different docking poses, we provide some particular velocities to the receptor and the ligand. And that particular velocity will try to rotate the bonds present in the receptor and the ligand. The energy associated with that is the delta G, the last one. The next slide, please. See, these are the couple of examples of the docking. See, here what is done is that the red one is the crystal structure and the blue one is the docked complex. What is done here is that we perform the docking and then we compare the docked complex with the actual crystal structure and check the root mean square deviations between their backbone atoms. And here, see, the backbone atoms are quite well superimposed. So that means our dockings are quite good. The next one is, next one, go to the next. This is another such example. Next one. This is another very vital example, but here see the red one and the blue one are quite different. That means they're quite separate. That means here the docking procedure failed. Generally, docking procedure fails for antibody antigen interactions. It is, I'm not saying this is a rule, but in general, it has been observed that docking simulation fails in case of molecules which have a very flexible region, and antibodies have flexible regions. The next slide, please. Here also, again, an antibody. So the interaction is not very good. That means the similarity between the actual structure and the docked complex, the structure of the docked complex is not very similar. The next slide. Here it is somewhat similar, but not here also it is not very similar. See, see, as I mentioned earlier, that docking studies are very good, but it is not always possible to mimic the exact biological scenario. It gives you a fair amount of idea, but not the entire idea of the entire process. And sometimes, definitely, it does not hold good always. Uh, but some of the portions you can find, they are aligned together. So those portions, you can consider those portions to be somewhat identical.
मेरा सच्ची
Okay, so this is what, in a nutshell, the docking procedure, the different types of dockings, and there are some real life examples. I told you earlier when I started that docking is a very complex process. Here, in case of computational docking, we consider only the receptor and the ligand. We do not consider, we cannot consider, I should say, the effect of solvent always. But in our cellular systems, the proteins and the receptors, they are present along with a particular solvent. So the solvents have very important influences on the conformations of the docked complexes. If we could find out a particular algorithm or a particular tool to dock two structures, considering the solvent effects, that would be a very good way of interpreting the actual biological phenomenon. But in most of the cases, unfortunately, we cannot use that solvent system in cases of docking studies. That is one of the most important drawbacks of molecular docking. But again, a ray of light is docking studies, molecular docking studies give you some amount of idea about how two substances, how a receptor and a ligand might interact. You cannot always perform wet lab based experiments. It is not always possible. In that case, using computational docking studies, you can at least identify the possible conformation of the complex. So that would definitely give you a fair amount of idea of the biological process that you are interested in, that you are studying. Okay, so that is the utility of molecular docking simulations. So with this, I'd like to conclude here, and I'd be happy to take any of your questions. You can ask me or you can write in the chat box.
Any question from the participants, you can write in the chat box or you can unmute and speak. Any questions? Please put in the chat box. Anybody is having any doubts in the docking? Please, you can ask to Professor.
Or you can unmute and ask me if you wish.
Question is there, will unpaid software like Autodoc give good results?
Autodoc definitely gives good results. But the thing is that you need to parameterize it. For example, say, first you need to take a complex for which the structure is known. And then that means the bound docking that I just mentioned, and separate its two components and let the Autodoc dock it in its own way. And then you need to check how close conformation you are getting of the complex from Autodoc. If you get a good match with the validated structure, with the structure that is available, which you used as your starting point, in that case, you can use those parameters for your future docking processes. That is a way to perform or to validate your results. It is always suggested to validate your results using some known information.
Any further questions?
Okay. Hello, Doctor. Thank you very much for a very good presentation. I have one question.
Yes.
There are many softwares available online also for docking, online platforms they provide. So how do we consider that which one or anyone we can choose for docking, or we have reliability, how we can consider that these are the softwares good enough, we can rely on them. So is there something, parameters are there available, or how should we choose? And the second one is, for a single protein also, there are many structures, right? So how can we choose the structure for beginning starting the docking process? So,
okay.
Can you answer, please?
Yeah, for your first question, yes, it is a really difficult task. As per my experience goes, if you have, if you can download a tool and if you can run it on your machine, it might give you good results, because in that case, you will be able to change the parameters, docking parameters. See, whether you are getting a good docking results or a not so good docking result, that depends on the parameters that are using for docking. There are different parameters. As I mentioned in my lecture, that you need to check the solvation energy, the binding interaction energy, all these interactions, all these software tools, they have some specified values of all these interaction energies. But those specified values will depend on the problem that you are trying to solve. Say you have a protein for which you have a large hydrophobic area. In that case, that protein might interact with its ligand using the van der Waals forces of interaction. In that case, you need to increase the value of the van der Waals potential. You may not have to use the one that is given in the software tool. So it is definitely a problem-specific question. It depends on the problem that you are trying to analyze. And for a particular protein, if you have a number of different structures, in the best one to be chosen is which one has the lowest value of lambda in crystallography. Lowest lambda value.
Okay, thank you very much, Doctor. Thank you.
And I think I have two more questions. Do we need crystallized structure of ligand receptor complex to validate? Yes, crystallized structure should be used, because NMR gives you an ensemble average. It does not give you a single structure. It gives you an average structure. So it is always good to have a crystallized structure. And how exactly can we find interactions between the receptor and the ligand? How exactly you can find the interaction between the receptor and the ligand? For that, what you need to do is you need to have some background information. You need to have the binding site information that I mentioned. That if you have the binding site information, that means which amino acids are present at the binding site. Say in your binding site, you have say glycine, you have say aspartic acid, glutamic acid, all these. That means your binding site is mainly negatively charged. So the ligand should be interacting with it in an electrostatic manner. That is an example. That is how you can find the interactions from the binding site. Yes, docking software tools. That is why docking software tools generate many different conformations. If you can specify in a docking process that, okay, I want the docking, I want the ligand to bind to this particular active site only, then your docking process, your docking method, your docking tool will give you results where you will find the receptor and the ligand are bound together in the active site only. But if you do not specify the active site region, in that case, you will get numerous results. From that, you have to pick and choose. Another question is, how can you identify the target for diabetes? It depends, like you cannot perform this with docking because you have to have literature. You have to know, you have to have this information from literature. Otherwise, it is not possible. Because docking studies, at least you have to have some background information about the receptor and the ligand. Is there any other question?
Any further questions from the participants?
Okay. Okay, thank you then, and good luck to all of you for your future research in bioinformatics. Thank you very much.
Thank you, sir, for your wonderful talk. Thank you so much.
Okay.
Yeah, we will now move to the next session. Okay, now I will be starting with the molecular modeling, and then I will cover the aspects of the docking. Docking apart from what sir has taught, I will be taking the other areas. So now, what is molecular modeling? So molecular modeling is a way of how we can predict the 3D structure of a protein. So from the linear structure, that is from the amino acid sequence, we can we need to generate the structure of the particular protein. So that particular process is called molecular modeling. So molecular modeling encompasses all theoretical methods and computational techniques used to model or even mimic. As Sir was telling that it is also, it definitely may not be the exact structure, but it will definitely model, or it will be, can mimic the behavior of the molecule. That is why the term here is called as model and not the exact. So computational method to model or generate the 3D structure of the protein. So molecular modeling is basically for around 3 types. One is homology modeling, other is threading, and the last one is ab initio. I think Sir has talked about this in the last classes. Still, I will be covering the much aspects of this homology modeling. So in homology modeling, what exactly it is done? You have the linear amino acid sequence with you. From the particular sequence, you need to search the structure whose sequence there is a match with the particular structure. If the sequence match is there, then it is predicted that the 3D structural match will also be there. So based on that sequence match, you are converting, or you are making the 3D structure. So that particular process is called homology modeling. Homology modeling is template based. So homology modeling refers to the constructing an atomic resolution model of the target protein from its amino acid sequence and an experimental three-dimensional structure of the related homologous protein, that is called as the template. So as I told you, a molecular modeling is of three types basically, and comparative modeling, actually the homology modeling and threading comes under the comparative modeling, because in both the cases you have the sequence and you are checking for the structure. In homology modeling, you are checking for the 3D structure in the protein data bank. In threading, rather, you are checking for the protein folds in the fold databases. And ab initio is directly from the sequence. You are modeling the structure without any knowledge of the template. So protein threading, also known as fold recognition, is a method of protein modeling which is used to model those proteins which have the same fold. Same fold in the sense, what are the folds like? They are alpha helix, beta sheets, and the turns. So the same folds they are taking from the particular 2D data, the protein secondary structure database, and that are assembled, and the particular protein model is created. And this particular
missing cases and details. പ്രത്യേകിച്ച് സ്ത്രീകളുടെ മിനിസ്റ്ററിന്റെ മകന്റെ കേസിലെ പെൺകുട്ടികളെ എന്നിട്ട് അതിൽ നിന്ന് 20-30 വയസ്സിനിടയിലുള്ളവരുടെ ഒരു ലിസ്റ്റ്.
Lavanya Delhi Babu, please mute your audio.
സാധാരണ ഭർത്താക്കന്മാരുടെ കള്ളുകൂടി, അതല്ലെങ്കിൽ വീട്ടിൽ സമാധാനം തരാതെ അടിയും വഴക്കും പോകണം, അല്ലെങ്കിൽ വേറെ സ്ത്രീകളുമായിട്ടുള്ള ബന്ധം. ഇതൊക്കെയാണ് സാധാരണ പറഞ്ഞു കേൾക്കുന്നത്. ഇതിപ്പം നല്ല അടക്കോ ഒതുക്കോ അനുസരണയുള്ള സ്വഭാവം. പിന്നെ എന്തിനാ മോളെ ഈ ഡൈവോഴ്സ്? എല്ലാവർക്കും സ്വന്തം അമ്മയുടെ സ്നേഹം അനുസരണയൊക്കെ വേണം. പക്ഷെ അമ്മ പറയുന്ന അപ്പോഴൊന്നും ഇല്ലാന്ന് പറഞ്ഞാൽ എങ്ങനെയാ? അതുപോലെ സ്വന്തം
Okay, go ahead.
So protein threading, also known as fold recognition, is a method of protein modeling which is used to model those proteins which have the same fold. As I told, the same fold, that is the helix, sheets, and the turns. And this particular and this is not saved in exactly in the protein data bank. It is saved in the protein secondary structural components. That is CASP, and CASP is there, and one more software is one more is secondary. There are two particular secondary structural information in that it is saved. Other one is ab initio. Ab initio is a method for prediction of the protein structures in the protein folding. So it is without any information or anything. Like in the homology modeling, you have the template information, and in the threading you have the fold information. But in ab initio, there is no information. Based on your own computational knowledge and the statistical knowledge, you are predicting the 3D structure of the protein. So again, I am coming to this homology modeling, and I will showing you some of the softwares. So homology modeling relies on the identification of one or more protein structures likely to resemble the structure of the query, and on the production of an alignment that maps residues in the query sequence to residues in the template sequence. So as I told you initially that in homology modeling you have a particular protein sequence, and its and its similar or homologous templates is selected from the protein data bank. So it has been shown that the protein structures are more conserved than the protein sequence among the homologs. Homologs means the similar protein sequences. So if at all it is like there is similarity in the protein sequence, definitely it will have similar protein structures. Steps in the involved in homology modeling. Actually, homology modeling is one of the most used method for determining a protein structure. Even if it is not the other option is threading, but usually homology modeling is one of the most widely used. Steps, the steps selection of the target, that is the unknown protein. Next is the selection of the template from the protein data bank. Protein data bank, you are selecting the template. That is the template is the one which has a structure, 3D structure deposited in the protein data bank, and whose sequence is similar with our unknown protein sequence. Next there is template target modeling or target template modeling. Next we have model building, and finally we have something called as model assessment. So once you are building the computational model, you need to validate the particular model. So that is called as a model assessment. The sequence alignment and the template structure are used to produce the structural model of the target. The quality of the homology model is dependent on the quality of the sequence alignment and the template structure. So everything depends on the target template alignment. And you don't need to worry on that, because there are softwares which are designed to do this particular job. So homology modeling by Swiss model. Swiss model is a particular server in which you just need to copy and paste the particular protein sequence, and the particular Swiss model server will be producing the 3D structure. So this is one. I have taken one model sequence, and the particular sequence is pasted in the Swiss model, and finally we will be getting the 3D structure in this manner. So shall I ask somebody to share the screen just for homology modeling? Who will be sharing it? Bhagyashree, are you there?
Yes, sir.
Yeah, I am enabling the screen share option. Please do it.
Yes, sir.
Yeah. Yes. Okay, and open any particular sequence, protein sequence from NCBI. Yeah, change it to protein. So you can select any protein sequence from NCBI on your choice. It depends on which particular protein sequence you want. There are, since as I told last day, NCBI is the so NCBI is the largest repository for almost anything, any biological sequence structures. Sorry, not structure, the sequences. Even you can select the amino acid sequence even from the EBI or DDBJ. Yeah. So since it is a server, it is very easy and you can do it. This is supported for the online classes. So and it is and it will be, since it is a part of EBI, it is easy to get it. Yeah. So click on the build model. It will be selected. Yeah. So you can wait for 1 or 2 minutes. It will be share. You can see the 3D structure. Somebody has put in the chat box as iTester. Yes, iTester is for threading. It will give you the rather it will give you the secondary structural components of the particular protein amino acid sequence which are submitted. So let this step run. You can unshare the screen. I will continue. Once it is generated, Bhagyashree, just let me know. You can again share the screen. Okay, it has done. Okay, good. It is coming. No, it is coming. Wait. Yes. So you can see in your right hand side, the 3D structure of the particular protein is generated, and you can just download the 3D structure. You can click that model 01, and there is an option like PDB format. Yes, you can download it, and then yes. Yeah, so it has got downloaded. You can view it in Rasmol or any other viewer. Other option in the right hand side, it is the JMOL. No need to open here. Word document. Try to open in this one Rasmol if you have. Otherwise, JMOL is there in your right hand side. Okay, click on the structure assessment. Yeah. Yes, below is the Ramachandran plot. So what is Ramachandran plot? I will explain to you very shortly. Very shortly. Yeah. Go back. And just go back. Yeah. Come little down. Come little bit down. Yes, you can see everybody. This model and template alignment. This is the target and the template alignment. So as I told you, the softwares are there to do this particular job for you. So already the particular sys model, it has selected a particular template, which is a template. You can see it is there in the red 4MBS.1, and it is having like 98% identity. So it is from the protein data bank. Yes. So that is how you have to assess. And there is some other mode of assessment also. So this is one more in which directly from the Swiss model you can do it. Okay, now you can unshare the screen. I will continue. You can unshare the screen. Okay, I will only do it. So this is a somewhat you had seen it. Here you can see this is the template and alignment. This is the alignment which you have seen in her in the particular website. You can see you have seen it. Now other one is this is for the threading. This is geno 3D. So in geno 3D you are just submitting the protein sequence and you are getting the secondary secondary structure information. You can see here this is PGP 2WDTNO. These are all actually the codes for the this one secondary this one for the secondary structural databases. So like this you will be getting it and you can directly model it. So like this you will be getting the models based on the geno 3D analysis and you can generate the 3D structure. So as like sometimes before Dr. Anshuman was telling, all the energy will be negative. It is minus kcal per mole. All the modeling energy and the docking energy, it will be in the negative. So here there is prediction for the Ramachandran plot. I will come to that. Okay, this is one software which is most widely used. That is Modeler. Modeler it is a software for generating the 3D structure. It is usually free. It is for the academic institutions. It is free. For the other non-academic, means for the personal ID, we will not be getting the particular license. But if you have this institutional ID, you will be getting this particular license of this modeler. So in this modeler, you have to submit the sequence and you have the option directly to select the target. In the Swiss model, there was that you cannot select the particular template. It is a software only select the particular template and it aligns. But in the modeler, you have since it is the software, you can download it and you can use it. You can take the particular template of your own choice and then you can align it and predict the 3D structure. So this is the alignment step for in the modeler. So this is how it is doing. In this is the particular protein and this is the template from the protein data bank. We are only doing it and alignment the software is doing it. This particular template we are only downloading from the protein data bank. The software is not doing it. Software is giving some of the choices. Based on the choice, we are downloading the 3D structure. You can see this is the alignment model, and wherever there is a star mark, these are all the perfect matches. And finally, the last step is model building. Okay, this is the 3D structure. Then next is the Ramachandran plot. I always mention in this particular software, it is initially in the biotech or bioinformatics, this is the only software which is done by an Indian. So that is why I always try to mention here and this is widely used all over the world and it is completely valid. So Ramachandran plot is based on the Psi-N5. It's called as the Psi-N5 plot. So this was originally developed in 1963 by G.N. Ramachandran, and there are some other people also. Mainly, he was the main Ramachandran. That's why it is based on his name. The particular plot is there. So Ramachandran plot is based on the dihedral angles, opposite dihedral angles. The first one is the psi and the next one is the phi. Opposite dihedral angles. Yeah, and one more thing I just wanted to mention here, proline and lysine doesn't follow the Ramachandran plot. Okay. So here this particular diagram I will try to explain to you. This particular deep brown region, this is a region which is the most accepted region, or the most favorable region, and the maximum residue should be concentrated in this particular regions. Then you have one more region, that is the light brown. This is called as the favored region, means maximum residue should be present here, and few residues can be also present here. So maximum residue should consider in this deep brown part, and little residue should be in the light part. Then the particular protein is very good. This is like additionally allowed region. So this in this particular area the residue should be minimum, and even in this particular white region also the residue should be minimum. Then only the model assessment based on this particular picture you can understand that which is particular protein is a good one or a bad one. If there are maximum residues in this white region and in the yellow region, the protein is not very good. The highest concentration of the residue should be in this particular region, then the protein is very good. Just by seeing the particular Ramachandran plot, you can understand where particular protein is good or bad. So you can see there are some residues. Yes, there will be few residues which can be here, but it should not be more. It should be very less. And you can see here it is given here in the bold like proline and lysine. Why it is mentioned? As I told you, it doesn't follow Ramachandran plot. Okay. So model visualization. Already you have seen in the Swiss model there was JMOL. Other software is RASMOL. So RASMOL is a particular program or a software. It is designed for viewing the molecular structure, the molecular 3D structure. So it is a molecular graphics program intended for visualization of the proteins, nucleic acid, and small molecules. Nucleic acid, but it should be in the PDB format. Then only it can be visualized. The program is aimed at displaying, teaching, and generation for the publication quality images. So you will be seeing almost a very good protein structure can be seen in this particular RASMOL. So RASMOL, this is a URL. You can download it and you can use it. RASMOL is free for any user. And this is the two things, that is hydrophilic plot and hydrophobic plot. Yeah. So hydrophilicity is a quantitative analysis of a degree of hydrophobicity or hydrophilicity. So this is particularly for the chemistry people. They will be knowing it what is hydrophobic and hydrophilic. It is considered to identify the possible structure or the domains of proteins. That is the likeliness or datingness of the water. That is the particular plot. The plot has amino acid sequence of a protein on its x-axis and the degree of hydrophobicity and hydrophilicity on its y-axis. There is a number of methods to measure the degree of interaction of the polar solvents such as water with specific amino acids. So this is the one. The top ones are hydrophobic and the below ones are hydrophilic. Okay, drug designing. So drug designing I will take in for you, but I will not touch that docking part, and I will go to some other aspect for the drug designing. So drug designing, as the name suggests, it is a particular process or it is a pipeline for designing novel ligands or novel drugs. So drug designing sometimes referred to as the rational drug design, or more simply, rational design is a inventive process of finding new medication based on the knowledge of a biological target. So once you know a particular biological target, example, diabetes. So the target for the diabetes you will be finding in the particular literature. So what is the particular target for the diabetes? So based on that, you can design the 3D structure of the particular target for the particular 3D structure of the target, and based on the structural information of the particular target, you can design the ligands for the same. So drug is most commonly a small organic molecule that activates or inhibits. That is, activates in the sense that it will be enhancing the function of the particular protein. Inhibits means it will limit the functionality of the particular protein. Example, if it is a cancer means it has to deactivate the particular functioning of the protein. That is why it is the inhibition function. And sometimes it will be having the enhancer function, like in the neurological disorders and all, it should be enhancer function should be there. So inhibit the function of a biomolecule such as protein, which in turn results in a therapeutic benefit to the patients. In the most basic sense, drug designing involves the design of a small molecule that are complementary in shape and charged with a biomolecular target with which they interact and they will bind to it. So drug designing frequently but not necessarily relies on the computational modeling techniques. You can do either way, either based on the computation technique or even directly in the wet lab. While you are doing in the computational platform, it saves a lot of time. In the wet lab, if it is completely wet lab, then it will be like more around 12 years to develop for a particular drug to come in the market. So this type of modeling is often referred to as the computer-aided drug design. Since we are doing with the help of computers, we are calling it as computer-aided drug design. And finally, the drug design relies on the knowledge of the three-dimensional structure of the biomolecular target. So based on the target only, the drug designing process happens. Okay, so typically a drug target is a key molecule involved in a particular metabolic or signaling pathway that is specific to the disease condition. So when we are selecting a particular target or the molecule, it is specific to the disease condition or the actions or effect of the particular disease. Or even if it is induced by any microbial pathogen, that also can be a particular target. So target selection mainly depends on your knowledge, and not only your knowledge, you have to refer the literature also. Some approaches attempt to inhibit the functioning of the pathway in the disease state by causing key molecule to stop functioning. Drug may be designed that bind to the active region and inhibit the particular key molecule. Another approach may be to enhance the normal pathway by promoting specific molecules in the normal pathway that may have been affected in the disease state. So docking, I will not go further. This is the particular figures that was explaining. So I will come to the other aspect of the drug designing. Okay, virtual screening. Virtual screening, the broader term for the molecular docking is virtual screening. Why the docking can be called as virtual screening? Because it involves so many processes apart from just docking. Docking is one of the steps for virtual screening, and above all, when you are doing a drug designing, there will be multiple molecules which will be interacting with the protein, and above all, you have to find some other functions also. That is why we are doing this virtual screening. And as you can understand by the term, it is virtually screened against the particular protein target. virtual screening is a computational technique used in drug design to search the libraries of small molecules. Small molecules, these are like you have so many small molecules. Example, databases like PubChem, Drug Bank, and so many other small molecule libraries. From there, you can select the particular compounds. Not only from the particular database, even if you are yourself designing the or creating the small molecule, that also it is possible. Because rather in the if you are doing a particular research, it is always good to take your own compounds and do the screening rather than taking the compounds which are already there in the database. Both ways are possible, both ways are research, but it is always good to do research on your own generated molecules rather than from the databases. So identify the library of small molecules in order to identify those structures which are most likely to bind to the drug target, typically a protein receptor or enzyme. So virtual screening, computational or in silico analog of biological screening. So it depends like on several factors like score or rank or filter a set of structures using one or more computational procedure. Which compounds to screen, that you will be knowing it, which library to synthesize, which compounds to purchase from the external source. Also used to analyze the results of the screening runs. Goal of drug designing, identify the ligand that tightly bind to the protein. Tightly bind to the protein, other way it is the docking, which the compound which are tightly docked with the particular protein, that you have to identify. Tightly bound, we will be seeing it within some times. Requirement, a computer database of the random potential ligands and the structure of target protein. Repetitive dock, that is the term repetitive dock, otherwise it is called as virtual screening. New ligands to the protein. Score, how tightly each ligand may bind, that is the docking score. Keep best hits, discard the other ligands. Keep best hits in the sense that which are the compounds which are actually binding and which are the ones which are exactly not binding. So you can see here, this is actually below is only one receptor, that is only one protein, and these are the ligands. So one ligand it is not binding, one this is binding, this is also not binding, this is also not binding. So definitely this is the best. Then this is the docked, this is not the docked ones. So you have to identify which is the docked and which is the best. Ways to assess the structure from a virtual screening experiment. Use a previously derived mathematical model that predicts the biological activity of the structure. Yes, either you can find out the previously designed mathematical model, means you should have a pre-knowledge of the model which predicts the biological activity of each structure of the protein. Run the substructure queries to eliminate the molecules with undesirable functionality. This is called as your pharmacophore modeling, wherein you are in pharmacophore modeling. It is usually of two types. One is structure based and other is ligand based. In the structure based on the 3D structure of the protein, not the 3D structure, rather the structure of the binding site, you are screening the compound or the ligand library. Other option is, suppose example diabetes, you have already medicines available. So based on the already available medicines, you are screening the particular medicine against the particular databases, and you are selecting the particular compound. That is what is called as pharmacophore modeling. So use the docking program to identify structures predicted to bind strongly to the active site. One thing is very important. There is one active site and one is binding site. It is not necessarily that all the binding sites are the active site, but active site is definitely a binding site. There will be other binding sites also present in a particular protein, and frankly, the particular compound it binds to the binding site, and it will be definitely, it may or may not be the active site, but it will be binding to the binding site. Filters remove structures not wanted in the section of the screening methods. Main classes of virtual screening depend on the amount of structural bioactivity data available. That is, one active molecule known performs similar search, that is called as ligand-based virtual screening. And 3D structure of the protein use protein ligand docking. One is ligand-based virtual screening, other is protein-based virtual screening. Yes, this is one important thing. Virtual screening methods for the non-specific targets. Prediction of the likelihood that a molecule has drug-like. So what is this drug-like characteristic and positive? This is a physicochemical property. This is called as a particular compound is it having drug-like properties or not. How you will be able to know it? There is something called as Lipinski rule of 5, which is it comes in the Cheminformatics, or it is a part of chemistry wherein you have to study the drug-like properties of a particular compound. So drug likeliness, so which feature of the drug molecule confer to the biological activity, analyze the value of the simple property, that is Lipinski rule of 5. Any chemistry person if it's there, that is better for them to understand. That is, poor absorption or permeation is likely when molecular weight is more than 500. Log P value is more than 500, that is measure of the molecular hydrophobicity. More than 5 H bond donors, sum of OH and NH groups. More than 10 H bond acceptors, sum of N and O atoms. This is the Lipinski rule of 5. Yes. So lead optimization can be done with the ADME properties, or the ADME properties, the other term is the Lipinski rule of 5. Say lead compound is defined as a compound, usually a small organic molecule that demonstrates desired biological activity on a validated molecular target. Lead optimization is accomplished through chemical modification of the hit structure, with modification chosen by employing knowledge of structure activity relationships, as well as structure-based design if the structural information about the target is available. Once promising leads are identified, they may go to the series of tests to provide an early assessment of their ADME T. There is one term, there is a slash here, ADME and T. This is absorption, distribution, metabolism, excretion, and other is toxicology. So usually it is difficult in a computational setup to predict the toxicology, but definitely we do this particular works. I will show you this 1 minute. Based on these results, most promising candidate enters the late optimization phase to make more effective and safer. At the end of the best form of performing compound is chosen as the candidate drug to enter preclinical development. So this is the next is the lead likeliness. So before that, I will be showing you this one, ADME. Example, I am selecting one particular compound, curcumin. So what I need from here, I need the Smiles code. So this mole inspiration is a software to predict the atomic properties of a particular molecule. So what you can see here, enter the Smiles or draw the molecule below. So you can definitely copy and paste the Smiles code and calculate the properties. So this is the anyway structure is generated. You will be getting the structure. So these are the properties. As you see in that molecular weight less than 500. This is the number of ON bonds, the number of OH NH bonds, and these are the number of atoms. And main here is N violation, means number of violations. If number of violation is, it should be 0. If it is not 0, this particular a particular compound will never have the drug-like properties. It should be 0. Even if it is 1 also, you cannot take it. Because any of the minimum, this 5 characteristics should be satisfied. That is why it is called as N violations, means the number of violations. Okay. So before docking or even after docking, you can do this ADME. Okay, so this is what is this ADME? So requirement for a drug must bind tightly to the biological target in vivo, must pass through one or more of the physiological barriers, that is cell membrane or blood-brain barrier, must remain long enough to take effect, must be removed from the body by metabolism, excretion and other methods. So admit absorption, distribution, metabolism, excretion and toxicity. Main thing is that if the particular molecule is able to penetrate the blood-brain barrier. So you have to check the permeability through the intestinal cells, membrane and blood-brain barrier. Based on it, we are doing in silico, but based on the in silico data, we can definitely go and tell that yes, it is possible. Hydrogen bond description, count the number of donors and acceptors, calculation of the overall propensity to be an acceptor or donor, modeling solubility, octane water partition, that is HON, that is the octane water partition coefficient and the blood-brain barrier permeability. Polar surface area, amount of molecular surface due to polar atoms, that is the N and O plus attached hydrogens, especially good for prediction of the oral absorption and brain penetration. And polar surface are greater than 140 square angstrom has been associated with the poor absorption. Description based on the 3D field. Molecular description quantify the molecule's overall size, shape and the balance between the hydrophilicity and hydrogen bonding. Toxic, actually it is, as I told, it is difficult to predict with the computer software, but yes, it is an important property, a very difficult problem. Most limit studies to single toxicological phenomena or single class of compounds, example polycyclic aromatic hydrocarbons, some based on the known toxic effect you are selecting. Okay, so virtual screening methods are central to many cheminformatics problems in design, selection, analysis. Increasing number of molecules can be evaluated using these techniques. Reliability and accuracy remain as problems in docking and predicting admit properties, and much more reliable and consistent experimental data. This is experiment, as I told, these are all in silico data. So now I want to show you the docking. Can somebody share the screen for docking? Saidipya is there. Can you unmute and you can show the docking? Saidipya, can you unmute, please? Or any other person who can demonstrate the docking?
Okay. I think they are not there. You do it.
Okay. Actually, they have the molecules. I don't have the molecules. They are already doing it. So I told them to do it. Okay. Okay, I am using the patchdoc server actually because it is not possible to run the docking software in my laptop. So as server setting, yes, it is always good to use a docking software, but due to several limitations, it is not always possible to download any software and do it. So since you can as the server setting that it is almost, and one more thing, the docking server's results will be it may vary, but final conclusion will be the same. Okay. So here you can upload the protein.
नहीं के पास डाटा है
Okay. So hopefully get the results. Okay, let me check if I have got the results or not. Some other. Yeah, I got the results. 1 minute. So there are so many confirmations you have got for this particular docking. So let me, so this is the docking score. Remember that you have to take it in the negative. If you given as a positive but nothing like that, you can definitely make it as -5506 kCl per mole.
अभी मुझे आप सबसे ज्यादा कॉन्फिडेंट आज नजर आए इतने हफ्तों
So this particular visualization, I am using the one software by name Pymol. So I hope everybody can see this Pymol viewer. So now you can see this is the protein ligand complex. How can you distinguish it? You can see that this is the interaction, this is the ligand, and there is the interaction between the protein and the particular compound. So hopefully everybody can see it. This is called as somebody was asking that sir, how to analyze the docking result. This is how you have to analyze the docking results. Now you can even find out that what is this particular compound. Sorry, this particular this one amino acid. That is also possible. The softwares run, systems get hanged very badly. So you can see here the interacting residues is LEU202. So this is the docking analysis. This one I have to remove it. It is not getting removed actually. It will not come actually. This is how the docking analysis you can do it. And you can even make this picture better. Okay, so I think you can all see this docking picture. This is the docking analysis. There is one interaction, and this is the interacting molecule. Okay, fine. So I will be ending the workshop here. It will have to be here. It's already time. So any questions you can please ask me. Please you can put it in the chat box. Any questions you can put it in the chat box. Any questions from the participants? Which code needs to be reported docking score? What are the factors you should consider for choosing receptor and ligand? That is the literature study. A docking score, this is the docking score. This is the docking score. Okay, which factors you should consider for receptor and ligand? That is based on your literature and how you are approaching. So if you have a particular disease, you have to find out what is the cause or the causative protein for the particular disease, and the ligand based on the property of the particular receptor, you have to select it. How to fix how to fix a target for the disease means you have to based on the literature, you have to find out the particular target, and you have to find out which is the receptor for the particular disease. Yes, any further questions? Any further questions? Okay, if there are no further questions, host can end the meeting. No, madam, you can end the meeting. Okay, then thank you so much.
Hello?
Indu ma'am?
Hello? Netra?
Yes, ma'am. Please end, ma'am. Sir has finished.
End.
Yeah, ma'am.
Yeah.